copying actually starts at a base we call 1. Alright? And I'm going to arbitrarily say that is here. This is where the copying is going to start. If we go backwards, the copying is going to take place in this direction. If we go backwards upstream, we come to two regions in the DNA. Something called the -35 region and something called the -10 region. Now this is not to scale. -35 means we start at that initial base and we count backwards 35 bases. Okay, that's what the -35 region is. And then for the -10 region, which we also call the Pribnow or Tata box, we would count back 10 bases. Alright? But for our purposes, I'm going to call the -35 region right here, and I'm going to call the -10 region right here, even though this is not to scale. It would actually be out here someplace like that. Okay? Alright. The player in this, in transcription, is something called RNA polymerase. RNA polymerase meets up with something called sigma factor out in the cytoplasm, and the sigma factor leads the RNA polymerase to the -35 region. So it's 35 bases upstream from where copying or transcription, copying is going to start. The sigma factor is kind of like acting like a seeing eye dog. It's helping the RNA polymerase find the DNA. Then the sigma factor goes away. The RNA polymerase moves down to the -10 region. Okay? When it gets to the -10 region, then the DNA starts to split. Then it moves down to 1. Okay? And when it gets to 1, that's where we start to get synthesis. Alright? Where it sees a T, it's going to put in an A. Where it sees an A, again, it's going to put in a U, keeping in mind that there's no T in RNA. All right. In my model, the DNA is yellow and the RNA is pink. And so to keep going along, where it sees a C, it's going to put in a G. Where it sees a G, it's going to put in a C, and keep going like that. And what's going to happen is the RNA, as it's made, is going to start to float off into the cytoplasm, okay, like that. Eventually, you reach a point in the RNA where it will backfold upon itself, forming something called a hairpin loop. That by itself can be a signal for transcription to stop, but sometimes it needs something called a Rho factor to come in as well. Okay? So again, going back to this is actually, if you're so fortunate as to draw this, this is actually a really straightforward one to do. So for transcription, sigma factor leads RNA polymerase to the -35 region. Then sigma factor goes away. RNA polymerase moves down to the -10 region. Then it will start to separate. RNA polymerase then comes down to 1, where it will start to perform synthesis of the RNA. Okay? And it will keep going along like this. The RNA will float out into the cytoplasm until such point as a region will fold back on itself, forming a hairpin loop, and that by itself, or with the assistance of the Rho factor, is enough to stop transcription. Okay? Questions about that? Okay. All right. So that is transcription. Again, that transcription is, very straightforward.